and foster discussions on effective solutions to mitigate marine pollution and protect our oceans for the future generations. With this background, we are organizing this seminar. For that, we have two eminent scholars with us today, one Dr. Vinith Kumar from NIOT and Professor Chandravadini Devadasan from Sri Lanka. The two experts will going to share their experience and expertise with us. For this, I am very happy to welcome our respected Pro Vice Chancellor, Professor Dr. Rajiv Balaji sir, to inaugurate this session. Sir is a very inspiration for all of us, and he is working very hard to promote maritime related domain as well as marine research activities. And we already have collaboration with the Eastern University on promoting marine science and maritime related research in Sri Lanka. With this background, now I am happy to welcome our respected Pro Vice Chancellor to inaugurate this webinar. Please, sir.
Yeah, namaste once again, Professor Chandravadini, Dr. Vinith Kumar, and the faculty of IMU. I'm very happy to be here. We've been having a series of lectures on various topics. The Center for Policy Research, as we had envisaged it, we wanted to become a think tank for the ministry and for the maritime domain, so that we can elevate policy-related talks and an academic flair to many of the topical issues. Marine pollution has been an issue for a long time, since the Torrey Canyon disaster, the Exxon Valdez, and so on and so forth. Oil was a major threat, and now we have other forms of pollution also, chemicals and the garbage and sewage, and also the plastic pollution. So it will be very topical to talk about these things, and I'm happy to see that the center is organizing more of these kind of lectures, and more experts are joining us. With these few words, I wish to express my pleasure that this session can be inaugurated. Thank you very much, Dr. Kodandaraman, for the kind words. Namaste.
Thank you, sir. Thank you very much for your kind support and guidance, sir. Thank you, sir. Thank you. Now, let us start the session. Now, I am happy to welcome Dr. Vinith Kumar, Senior Scientist at NIO, National Institute of Ocean Technology, and I would like to give brief introduction about the speaker. Dr. Vinith Kumar has completed his Master and PhD at the CAS in Marine Biology at Annamalai University. After that, he joined as a scientist at NIOT, National Institute of Ocean Technology, and presently is a project director at NIOT with an extensive academic and research background in marine sciences. He is leading a deep ocean mission Vertical 6 advanced marine station for ocean biology. His areas of specialization include marine bioinformatics, marine ecology, and biochemistry, where he has research focuses on understanding the complex of interaction within marine ecosystems and utilizing bioinformatics tools to address the critical oceans-related challenges. He has vast experience and expertise that today he is going to deliver a talk on how this marine biotechnology research is evaluated and the significance as well as the progress in India, and how these marine biotechnological tools can be utilized to minimize or protect our marine coastal ecosystems. With this background, now I am handing over this platform to Dr. Vinith Kumar. Please, Dr. Vinith Kumar.
Yes, sir. First of all, I would like to thank Dr. Chandravadini for contacting me first, and I got the approval from director, and then IMU has contacted, and thanks for IMU for coordinating and organizing this webinar. So I would like to first say good morning and vanakkam to all. So right now I am working as a scientist in Marine Biotechnology Group of National Institute of Ocean Technology. This National Institute of Ocean Technology is generally it is an engineering group, engineering related institution. So our main focus is on developing engineering related that is various technologies and providing solutions. And apart from that, harvesting this, harnessing this living and non-living resources. This is the major aim of our NIOT. Anyway, with that, this thing I was working, I have joined in NIOT in the year 2000. Then from 2005 to 2023, I was, in 18 years, I was working in Andaman Nicobar Islands, developing a Atal Center for Ocean Science and Technology lab. There we were doing a lot of research activities. My presentation is, then after that, I joined here and taking care of this in that Deep Ocean Mission, Vertical 6, that is Advanced Marine Station for Ocean Biology. So this is one of the ambitious program. So I will give all the details about our activities, research activities, which is particularly happening in the areas of marine biotechnology and even in pollution monitoring, what we are doing. All these things I'll give us a overall view of our NIOT activities. And also I will just introduce about our NIOT and also Deep Ocean Mission program, which is being carried out by Ministry of Earth Sciences through various institutions of Ministry of Earth Sciences. The major institutions is NIOT, which is operating 4 verticals. So I'll share my presentation. Can you see this in full screen?
We can see this, but not full screen.
Just a minute. Can you see this now?
Same, sir. We can see your presentation, but
yeah, okay. Then I will share it in different mode. Can you see now?
Yes, sir.
In full screen?
Yes.
Yeah, thank you. So this National Institute of Ocean Technology, which is under the Ministry of Earth Sciences, and the head office is located at Chennai, and there is one more center at Andaman Nicobar Islands. Apart from that, NIOT is having various field offices at Lakshadweep and other places, and doing a lot of different projects. One of the notable work is in Andaman Nicobar Islands. We have a center, and we are doing all marine biotechnological studies as well as pollution monitoring, and we are doing environmental impact assessment surveys. Apart from that, various services we are providing to the islands. Then apart from that, particularly in the case of Lakshadweep, we have installed desalination plants. So the people in the Lakshadweep Islands, they can drink the water which is produced by plants by NIOT. So this is about this one. So as I said, this main aim of this NIOT is to develop reliable indigenous technology to solve various engineering problems associated with harnessing non-living and living resources in the Indian exclusive economic zone. There are 3 sub headings also. In that, our main aim is to develop world-class technologies and application for sustainable utilization of ocean resources to provide competitive value-added technical services and solutions to organizations working in the oceans, and develop knowledge and institutional capabilities in India and management of ocean resources and environment. So through this, we are collaborating with the different institutions and all over the country, India, and also in the various institutions located at abroad. We are providing solutions through various ministry institutions. The major programs of NIOT include coastal processing and modeling, hydrographic, geographical surveys, offshore structures, mechanical engineering, and one of the program is Ocean Science and Technology for Islands. This is the only the biotechnology group which is working in NIOT. Under that, the Atal Center for Ocean Science and Technology for Islands, which is located at Port Blair and Port Blair, that is now Sri Vijayapuram at Andaman Nicobar Islands. Other than that, there are technology projects like deep sea submersible, deep sea mining, desalination, gas hydrates. Then other than that, we have operational program like vessel management cells. We have 4 ships. Two of them are going in the high seas, and Sagar Nidhi, and there are other vessels, Sahar Anvesiha, that will go to the coastal areas. Then apart from that, we have ocean observation system. So we deployed various kind of data buys in the offshore conditions, and also Argo floats, which will collect all the information. It will send to NIOT through satellite, and this data are processed for cyclone warning and all these things. We have a very robust network for this. And apart from that, in the year 2021, the Deep Ocean Mission is also added. Under that, many programs are now working, under the Deep Ocean Mission. So, uh, where the major program in Andaman Nicobar Islands, as well as our group in MBT, is open sea cage culture, marine algal biotechnology, marine microbial biotechnology, island resource information system, seawater quality monitoring along the Indian coast, and development of potential drugs. That is various microbial products we are developing, EAA, and other support to other engineering groups. And now we are coming to Deep Ocean Mission. This is the major program being carried out by NIOT. This program was launched in the year 2021. Then it was supporting to the marine industry to enhance the blue economy. So the total cost of that mission was 4,000 crore, and presently the first phase is going on with 2,823 crores, and the second phase will start after this. This first Now we got the approval of extension of this first phase itself. Due to COVID and other reasons, there were some delay, but now it is going in a very full swing. So there are about 6 verticals in this Deep Ocean Mission. Under that, first mission is.
Sorry to interrupt. Your presentation is not visible. It is
Oh,
was visible at a full screen. Now I don't know what happened.
I'll share again.
Yeah, please.
Can you see it now?
Yes, sir. Now it is visible.
Yeah, previously just I was giving about some information about NIOT and
Yes.
whatever I talked about. Now we'll talk about Deep Ocean Mission. The first vertical is being operated at NIOT. This is submersible we are going to develop. This submersible go up to the depth of 6,000 meters, and this can carry 3 people for exploration of underwater non-living and living resources. And this is and apart from that, we are also developing polymetallic nodules mining systems. That is deep sea crawler also we are developing. This is the major aim of this first verticals. So so mining technology and submersible is the major one, this first vertical, which is being carried out NIOT. The second vertical is Ocean Climate Change Advisory Services. This is being done by INCAIS, Indian National Center for Ocean Information Services, which is located at Hyderabad. They are already giving potential fishery zone forecast also. Apart from that, they are going to predict the model for different climate variations, all these things. And this is they are also deploying these Argo floats, and they are collecting the informations and data from our own ocean deep sea database. From that, they are predicting the that is sea surface temperature, as well as they are even giving cyclone warning along with Indian Meteorological Department. They are working in coordination with the NIOT and other institutions for all these purpose. The third exploration, third one is third vertical is exploration and conservation of deep sea biodiversity. In this, three institutions are participating. One is NIOT, another one is Center for Marine Living Resources and Ecology, which is located at Cochin, and another one is BARC, Baba Atomic Research Center, Kalpakam. So NIOT's part is deep sea microbial taxonomy and genetic studies. Other that is CMLRE, Center for Marine Living Resources. They are doing all kind of invertebrate, vertebrate, all macroorganisms they are studying. That BARC is doing on biofouling studies, particularly for marine environment, and developing technologies for anti-fouling coatings and all. This is so. This has three aspects in this vertical 3. Vertical 4 is about deep ocean survey and exploration. This is being carried out by one of the institution of ministry, that is NCPOR, National Institute of Polar and Arctic Ocean Research, which is located at Goa. They have separate ships, and they do a lot of services that surveys in the Indian Ocean, offshore, Indian EEZ and other places. So they are going to have separate vessels also for this. Then fifth vertical is being operated at NIOT. This is energy and freshwater from the ocean. This particular one is very important in terms of ocean thermal energy conversion. Initially, it was demonstrated. Now, land-based system also, land-based and offshore-based system, they are going to develop. And already based on this technology, we have already deployed in 6 different places desalination plant in Lakshadweep. So they are producing more than 1 lakh liter of water per day, which is sufficient to meet the daily requirement of the local people in small islands like Kavaratti, Minikai, and all these things. Then sixth vertical is development of this advanced marine station for ocean biology. So each vertical has project directors. For this sixth vertical, I am working as project director, and we are working in under the Marine Biotechnology Group, which is headed by Dr. Dharani. He is also the project director for vertical 3 of this program, Deep Ocean Mission program. So main aim of this advanced marine station for ocean biology is infrastructure development. We have already purchased land. This is going to be a hub and spokes model. And there will be different labs and also virtual station. We have already developed our website and dashboards, and we'll have that all different kind of research in marine biology, marine ecology, and capacity building. We are going to provide postdoctoral and PhD and fellowships, and also we are starting marine biology courses in different universities, MSc marine biology courses, and we are supporting their students, some 25 students with fellowships. Apart from that, PhD and postdoctoral fellowships, 150 numbers each, we are going to give through these vertical six. And we are internationally collaborating with French and other France and other Germany. And apart from that, we are trying to collaborate with East Asian, South Asian institution also. Then we are also interested in business development, particularly in case of incubator facilities. This is the overall this one how this marine biotechnology activities being that is we are doing initiating with our this thing. Then what are the activities we have done in this, we will give a detailed report. So this is the website amsob.in. You can go there and we can see what are the activities being carried out. Apart from that, you can visit the NIOT site and you can find out a lot of activities. So under the vertical 3 deep ocean mission, we are collecting samples from our EE set in different latitudes, and so our particularly our we want to at least cover 10% of our coastal area for to identify this piezophilic and piezo-tolerant. Piezo-tolerant is bacteria. Actually, piezo-tolerant means they are tolerant to pressure, and piezophilic they are living in the pressure realm of deep sea. So we will collect not only the microbes, we will also collect microbes available in the water and sediment column. Apart from that, which are also available in the symbiotic relations with the organisms. These also we are studying from our EE set, and also beyond our national jurisdiction. That is biodiversity beyond natural jurisdiction. That is BBNJ. Based on BBNJ agreement, also we are doing these studies, and we have not now carried out more than some 40 that sites from in India. So for studying this, we have already developed separate that is water sampler as well as pressure retaining culture facility. This water sampler also will go up to 6,000 meter. We can go and collect the samples, and they will retain the pressure and temperature of particular conditions in the deep sea, and we can bring the samples and transfer to this particular system, which will retain the temperature and pressure, so that the bacteria or microbes, other fungi and other microbes living in that particular depths will produce the bioactive compounds, which is important for that particular depth. For example, some of the bacteria which are even tolerant to the surface atmospheric temperature, atmospheric pressure. They can be cultured. They are called piezo-tolerant. They can be cultured in the normal condition, whereas piezophilic organisms, microorganisms, can be cultured only through this system, which is developed it is available only in NIOT in entire India. So we have done a lot of work, and we found out some of the bacteria which are producing various bioactive compounds, which are in that particular temperature and pressure conditions, which cannot be produced during in the normal atmospheric conditions. So another thing, these pressure tolerant bacteria also will not survive in the normal temperature and other conditions. They will work in that particular temperature and deep sea conditions only. So we are mimicking that deep sea conditions in with this our this is specially developed systems which is available in NIOT, and we are working. And apart from that, we are developing sampler also, deep sea sampler indigenously we are developing. This sampler also not only sample bottles which will retain the pressure of pressure as well as um, this temperature conditions. You know, every that 10 meter there will be 1 atmospheric pressure. So at 6000 meter we will get 600 atmospheric pressure, which will be very high for this bacteria. And when these organisms are coming to, sometimes this even invertebrate and other organisms are coming to surface, they will bulge, and they will because of atmosphere their pressure difference, they will die. So whereas these bacteria, pressure tolerant bacteria can be cultured, where that also pesophilic bacteria can be a culture. This is one of the special work we are doing. A lot of studies we have done. We have made we have done some patterns also. So these are all the sampling sites we have gone for that is cruises in that up to 2000 meters, 2 to 3000 meters we have studied the collected the samples. And apart from that, there are unique ecosystem in the offshore conditions. They are sea mounts, underwater sea mounts. They have separate. They may have sometimes hydrothermal vents. That hydrothermal vents has separate organisms which are living in the higher temperature conditions. Recently, also our NIOT has found out some of the hydrothermal vents in the southern, that is Andaman, near south of Andaman. And they also found out some of the polymetallic nodules in the 1200 meter depth. They have collected polymetallic nodules, and we are also studying that bacteria and other microbes associated with these polymetallic nodules. So because that not only this through this uh, for this many of the Indian institutions are collaborating with us through Deep Ocean Mission. We have given funding for different institutions all over India for collaborating for biotechnology, as well as each verticals we have given funding. For vertical 3, that microbial biodiversity and advanced mineralization, more than 40 projects are given. We are sharing the samples with this institution, and they are working on various deep sea bacteria, viruses and fungus, and many other kind of microbes. They are working on that. So these are all the deep sea microbes, some of the microbes which was isolated from deep sea conditions, and these are all pressure tolerant. Some of the thing which you can see in the right side in the Petri plate, these are all pressure tolerant bacteria. Some bacteria are which are pesophilic. That is, they are living only in that. They will survive only in that pressure condition. Then more than 230 bacteria we have isolated and found out for carbonate dissolution activity, and like this carbonic anhydrase activity and extra polymeric substance production. Then apart from the polyaromatic hydrocarbon production, degradation potential, like that, then more than there are some bacteria which are of biosurfactant nature. So these are all from the deep sea organisms that particularly microbes we have identified and studied. And not only we are not only studying the bacteria in culture dependent and culture independent bacteria also we are studying. You know that all the bacteria which is available in the say that deep sea are in the surface. they cannot be cultured. Less than 1% of entire bacteria can be cultured. So remaining, we have to study through eDNA aspects or to metagenomic studies. So in our NIOT, both works are being carried out. We have a lot of metagenic genomic clones, more than 400 bacteria. We have metagenomic clones that can be studied by different group of people. Many people are approaching us. They are taking these clones for further studies. So there are so many bacteria which are available in the deep sea condition. We are studying them in our institution with deep sea institute conditions. So under 10 bar or 20 megapascal that pressure conditions. So one of the bacteria, Brucella anthrophi, which are also studied for different pyrene degradation and many other studies. There were from this bacteria we could able to find more than 4,419 proteins. Particularly this genome sequencing done for this bacteria, and we have annotated the sequences with various bioinformatic tools, and we also found out various xenobiotic and biodegradation potential of this organism by studying their metabolic activities and metabolic metabolism also. So through using KEX analysis, that is particularly this software, it will be KEX. KEGG software will be used for this metabolism studies. Then apart from this transcriptome also of these organisms also used for various studies. Then other culture independent bacteria, this using metabolic studies, if you see that there are 40 metagenomic, this one we have studied, and these are all available for collected. The samples are collected from water as well as sediment, and there are various group are representing based on the metagenomic studies, from starting from Omnitrophia to, and there are so many unclassified bacteria which may be new, or we have to find out, or we have to give the names of identify these species. This is very enough new to our studies, because this has a lot of potential for studying the genomes and useful genomes or genes which will produce various bioactive compounds. We can identify these bioactive compounds for different purposes. So metagenomic studies are one of the very important this thing. So apart from studying this bacteria, we wanted to keep them under pressure, deep sea pressure conditions, and also we want to store all these microbes. For that, we are developing microbial information, that is facility, as well as we want to have this microbial repository. So at we have another center near our Chittedu, Andhra Pradesh. We have 200 acres of land. There we are constructing a big lab with ground plus one floor facility, where that lot of these microbes will be stored for different people can approach, and they can collect these microbes for various studies. We have visited various microbial culture collection facilities located all over India, that is INTEC, Chandigarh, NSS Pune, and NCL. And apart from that, we are going to visit international that culture collection, bacterial culture collection facility also, particularly in Germany and France and all. Based on that experiences, we are now developing this culture collection facility also, repository also. Then apart from that, already we have developed a marine microbial information portal, which will be available, MMIP, which will be available soon for for public use. This not only we can collect all this microbial genomic information, which can be stored in this microbial portal. Particularly, this is for Indian microbial portal. Generally, that is NCBI and other international microbial that is genomic um, that is portals are collecting information from land as well as marine and various realms. But in this case, it is only marine microbes, so that we can find out the various product which is being segregated by this microbe, and also it can be used for various studies. And this also this various pipelines, through those pipelines you can study the functions of this particular microbial genomes, and you can do simple blast and many other tools we are developing through this marine microbial information facilities. Then apart from that, we have done a lot of that biotechnological this one, in particularly plastic degradation bacteria. Some of the we have developed this technology, and this technology is with this technology you can degrade low density polyethylene, that plastics. Within 120 days it will reduce 48% of the this LDPE. Similarly, we have also studied this mercury bioremediation by deep sea microbes. These deep sea microbes, some of them collected from the central Indian Ocean, which are all having MER-A genes, which are responsible for degrading this mercury compound into that is into mercury H2 plus into Hg, that Hg0. So this mercury will not be stable in that environment if it is contaminated area. When we expose these bacteria to this one, they will degrade the mercury, so that mercury will not be this elementary mercury, particularly Hg. They will not stay in that environment. They will get evaporated from that site. Thereby, after sometimes or particular days or something like that, mercury will not be available in the contaminated site. This consortium of bacteria also we have developed, and this we are going to develop as a technology, and this soon it will be coming to the use. Apart from that, we are also doing a lot of marine microalgal biotechnology. More than 225 strains of we are keeping in our NIOT, and they are producing various bioactive compounds like lipids, fatty acids, and many other that is pigments also. So we have developed a technology, and for mass culture of this microalgae using bubble column reactor and tubular photobioreactor, and also raceway. This raceway also located at that is Pamanji on Andhra Pradesh. So we can develop this microalgae, and like chlorella, spirulina, many other microalgae can be used for biomass development. This microalgae particularly, which can be enriched for that is fatty acids or other compounds. This will be used in development of feed for particularly shrimps and fishes. This can be added with that while we are making that feed for these fishes. And some of that microalgae are used for human consumption. Apart from that, there are a lot of nutraceuticals which are available from the microalgae. These are all we are using with different bubble column reactor and with a raceway. So we can find out within short period, 5 to 10 days, they will be able to, we can able to produce the large quantity of biomass, and which can be used for this one. In our laboratory, we have used Chlorella vulgaris, Chlorella soracniana, and many other species. And we got a very good amount of lipid from this. So these are all the. And then apart from that, harvesting is the biggest problem for this harvest, this microalgae. You can culture this one since they are microbial in nature. They are, we have to use centrifugal force or something like filtration like that for that will be very expensive and it will be time consuming. Yeah, so NIOT has developed electroflocculation technique. With this electroflocculation technique, we can harvest more than 98% of this algae within a short period of time. Within few minutes, they will float in this kind of flocculation will happen, and so that harvest. This microalgae can be used for aerofuel development as well as biodiesel production. So some of the countries, they are producing using this microalgae. They are producing a lot of biodiesel. Apart from macroalgae and other plant biomass, this is one of the very good sources. And we could able to extract, and we can run the vehicle for more than some 120 kilometer. So initially we have done, but for large scale commercial aspects, we have to do a lot of experiments. So apart from that, one of the important compound which was isolated from microalgae is lutein, which is very costly, which is used in pharmaceutical and many other that is areas. This lutein also we have isolated, and we have developed this technology and transferred this technology to other that particular companies. Then apart from that, we also isolated and extracted this phycocyanin from marine spirulina. This is one of the very important compound used for many other that is pharmaceutical and purpose. We use different techniques for extraction, particularly this in this case ultrasonification we have used, and thermal and these particular spirulina based this phycocyanin is actually thermally stable. So in other cases, it is not so. So there are a lot of use while we are doing microbial, that is marine microalgal studies. Apart from that, we do seaweed culture also, seaweed biodiversity also. These are all some of the seaweeds which were isolated or identified from Andaman Nicobar Islands, and this one. So this is seaweed culture. Initially, we started with the bamboo wraps, and we used mostly Gracilaria edalis, and this Gracilaria edalis, which is available in Andaman Nicobar Islands, but in Andaman Nicobar Islands, mostly these alginophytes, particularly sargassum and other species, are available, terminaria, in high quantity, whereas Gracilaria is in limited quantity. So we have to culture them. So initially we started with the bamboo raft culture. Later we shifted to PVC rafts. When instead of individual that smaller 3 into 3 meter bamboo raft, which will not stable for a long time, so we shifted to PVC culture. Then also we found that PVC also degrading. So we used, now we are using HDPE, high density polyethylene tubes. That is, this is long line method. With this, we can go in a, we can develop that Gracilaria, even Kappa ficus we can culture in Particularly Andaman Nicobar Islands, we have not promoted the Kappa Pike Salvarisi. But in Tamil Nadu, particularly in Mandapam area, Rameshwaram, apart from that, Gulf of Mannar and Tamil Nadu East Coast and Gujarat, Kappa ficus and are being cultured. Apart from that, there are species, Gracilleria dura, all these things are being cultured in different this thing. In Andaman Nicobar Islands, we started with Gracilleria at least, and we could able to go up to initially with a few kgs, 10 kg, 15 kgs. Now it is going up to 1.5 tons. We could able to culture. Now we are going for offshore culture. Some 1 hectare land, 1 hectare water area, we will be culturing this one. So we are for this we are associating with a private we are going to associate with one of the important company, which is Indian company, which is doing that seaweed culture at Indonesia in a large scale. So this is the luxuriant growth of this Gracilleria adelis. Particularly this Gracilleria adelis and other species are very good for production of agar, which is being used for all the microbiological laboratory. And agar seaweed is the only source for agar. No other source is available in the world. Seaweed, it is all collected, either naturally collected or cultured. So these are all the methods we have developed for long life. Now we are going with HDPE, because HDPE advantage is yeah, you can.
Dr. Vineet.
Yes, slide is moving.
No. One of our faculty want to have some clarification. Can you answer now or end of the session?
Yeah, no problem. We can answer now.
Yeah, please, Dr.
Yes. Good morning to everyone. I just want to know that this microbacteria which is found from the sea, they are also useful for generation of hydrogen energy, hydrogen gas.
Yes.
So have you got any kinds of species which is specialized for that?
Actually, we can work.
Indian Ocean?
We have given some project to some different people. They are working on that. And hydrogen energy, we in our lab we have not studied, but deep sea bacteria are able to produce some of the bacteria they are producing. We have to do some experiments. So some of the people are doing it. So one project is given in this related to this hydrogen energy production.
Okay, and another questions are like you have explored like in the ocean depth of 6,000 meter. Did you get any precious metallic substances in the Indian Ocean, like something like which is very having high value and important to country?
Yeah, that is what that our polymetallic nodules mining is being done by our 6,000 meter Indian Ocean also. Our people have done some previous studies. But the thing is the extraction of those this one that polymetallic nodules from deep sea depth to and bringing them to the surface. So there NIOT is working on those technology, deep sea crawler and pumping systems that is being worked out. And apart from that, we have demonstrated up to 500 meter depth. This slurry is being that is collected and it was transported to surface vessel which is located in the surface like that. But deep sea condition at 600 bar pressure, it has to be demonstrated. No other countries are having that kind of systems, and one or two countries trying, and NIOT is also trying in our level best. And in southern Indian Ocean, particularly in the Andaman Nicobar Islands, south of Andaman Nicobar Islands, our people have recently collected polymetallic nodules in 1,200 meter depth itself. So we feel that our that deep sea crawler system can extract those this one, but in simultaneously we are looking into that environmental aspects also. How much, what kind of environmental effect will be there, all being studied along with this crawling systems also. we are doing all this thing, how to minimize and mitigate all those effects. Those are also being carried out.
Okay,
So
sir.
we are studying.
One more question, that your research, when you are exploring the sea, uh, is it also includes the, let's say, exploration of and getting the new oil field,
No,
methane
oil
gases and other things?
exploration.
Or it
Oil
is
exploration. Yeah, oil exploration is a separate part that is being done by ONGC. So NIOT is not involved in oil exploration kind of things, because there is a separate department, and they have separate equipments for drilling all these things. NIOT is not doing on that oil exploration related things.
okay. One
But
more
we are
question.
working on gas hydrates. Some places gas hydrate where at what depth available. That that kind of studies only NIOT is involved.
Okay, any of this species which you got in the micro, this bacteria, do they have a lot of mediclinal values also, like medicines value, etc.?
Yeah. I'm going to give you the that other slides. It is going to come. Some of the patterns we have transferred. There are a
Thank
lot of things that, but as we have developed mostly nutraceuticals, because as drug, if you want to market it or something like that, it will take at least 20 years to bring a market, to bring to a market, because it requires a lot of testing and all. So drugs we are collaborating with the Drugs from the Sea program. They have brought out some of the CDRI, Central Drug Research Institute, Lucknow. They are working on that along with us. The samples were collected from Andaman Nicobar by our group, and we have given to them, and they are now working on that particular aspect, and they are doing it. And they have brought some of the products for as after testing with the different this one. So for example, human trials, all these things need to be done. So that is not that's why we mostly concentrate on that, particularly nutraceuticals. As well as drugs compound can be there are a lot of potential drug compounds we have. So
So
it
I
will.
think that you might have registered some patent also after getting some new
Yeah,
bacteria for very valuable bacteria from
yeah, I'm coming.
NIOT.
I'll show you in the next slides.
Okay, okay. Thank you.
Yeah, thank you. Yeah. This is, can I continue that?
Yes, sir.
Yeah, actually, this is one of the long line method. We are doing it in Andaman Nicobar Islands for using HDPE. The HDPE, as I said, it will last for even 20 to 30 years also. There will not be any problem. Only ropes will be, you have to change after sometimes 5 to 6 years after that. Otherwise, this nets and everything. So you can minimize the pollution in that area. Apart from seaweed culture, we are also doing lot of products from the seaweeds, collecting lot of products from seaweeds, particularly biostimulant, which will plant growth promoters, and also bioplastics from the seaweeds. This plant growth promoters, which has lot of gibberellic acid and kinases, other plant growth hormones, which will help for fruiting and shoot and plant growth, and which these are being tested in different agriculture crops, and we could able to get good crops out of that. Even tested in different like pumpkin, lady's finger, tomato, even in rice paddy field we tested. We are getting the good results, and particularly these are all extracted from some of the seagrass which was found in Andaman Nicobar Islands. Already there are a lot of seaweed based biostimulants are available in the market. Many of them are European based Ascophyllum nodosum based bioproducts. But in India also we have a lot of seagrass and seaweeds. They are equally good and producing. They are marketing well, but we are our marketing also initiated by our local this one companies. So they are doing it well. Then apart from that, we have also produced these bioplastics from seaweeds. Some of bioplastics are edible bioplastics, which can be used for packaging of food and other materials. Some are only for packaging. Two kind of bioplastics we have produced from that. So these are all the technology transferred. I want to tell you some of them are anti-cancer enzyme, anti-cancer enzyme, L-asparaginase biosynthesis gene. We have actually this L-asparaginase which is available in the market, which is already available in the market. But the thing is, it is very costlier, and also this particular L-asparaginase, which is used for cancer treatment, we isolated this asparaginase from one of the deep sea bacteria. Deep sea bacteria. This will be actually. This is also actually while extracting this L-asparaginase, sometimes another glutaminase is coming as a contaminant through this, which is also not good for human health, which we can have, it is very difficult to separate. But this L-asparaginase, which we isolated, which is pure and it is better, having better activity, we have transferred this gene to another bacteria through gene cloning techniques, and these clones we have transferred to other private companies through National Research and Development Board, New Delhi, that they are the agency who are transferring to different this is they are particularly the NRDC's main this one is technology transfer. So we have given this sample to different companies, Derma Pharmaceuticals and other places. Apart from that, some Derma Pharmaceutical by Ectoin, that is this Ectoin is osmoregulant, and also they can use used in skincare products. This also we have through transferred this gene containing clones to the different companies they are working on. Then apart from that, we have developed a multiplex PCR kit for actually detection of enterococcus bacteria, which sometimes these pathogenic enterococcus, they can produce diarrhea and all. Normal conditions, if you want to test the bacteria and isolate, test the bacteria, it will take more than 48 hours to identify bacteria. But with this kit, you can identify with the single kit itself, you can identify the bacteria within 3 to 4 hours. So treatment can be immediately started. Apart from that, there are other biosurfactant marine bacteria which can clean the oil, particularly oil, degrade the oil, and so we can use it for environmental cleanup. This also one EcoBuild Corp Private Limited, they have taken it from sir. Then apart from that, as I said, Ectoin, this also one Cosmos Biotech to taken. These are all transferred through NRDC only. Then this Ectoin itself, 2-3 companies they have taken that. This one also this L-asparaginase also there are global hospitals and other places they have taken. This is very few I am telling that technology we have transferred. Apart from that, I am providing only 5 patents we have done for this Enterococcus multiplex PCR kit. Then this microbial harvesting system. That is then another. It is European patent and German patent, Indian patent, other things. Then there are so many other things. There are another 10 to 15 patents done from our laboratory. That we are also into the open sea cage culture also. So in this is being done in Andaman Nicobar Islands. So actually these 9-meter dia cages, initially we have actually imported from Turkey. Later we indigenously developed all these things in India. In India itself, now we have suppliers. We gave them the technology how to prepare the molds and nets, all these things they have produced. And These are all the mapping we have done for where and all that cage culture we can do. What are the suitable area, what are the not suitable area. We actually tested our cages in different environment, protected environment and semi-protected environment and open sea cages. They are stable, very much stable. And we have conducted a lot of training for this. Apart from that, we are also developing submersible cages. These submersible cages, when there is cyclonic conditions or something, they will go down inside the water. When the rough conditions goes, it can come up. So we can it will go down along with the cultured organisms, particularly cobia and sea bass. These fishes are technologies are available. We have also NIOT also developed that underwater feeding that is systems. So even in the subsea conditions, we can provide feeding from the top itself. So this is another thing that now we have developed different kind of technologies for that. These are the harvest we have taken. So from different places in Olaikuda, that is particularly at Rameshwaram, Tamil Nadu, then Andhra Pradesh, and various tons of 6.5 tons, 4 tons, like the different species, particularly sea bass and cobia. Cobia actually they have a very good growth rate. Within 8 months, they can grow up to 4 kg, whereas sea bass will grow up to 800 gram in 8 months or something like that. So the profit will be very high for that cobia culture. So apart from that, there are other fishes also we have cultured, parrot fishes and milk fishes. So many places we are doing this one. In Andaman Nicobar Islands also we have demonstrated this culture, and even now it is going on. Culture is going on. Apart from that, we have developed different artificial reefs and deployed it in Andaman and other places, Tamil Nadu coast, in different places. This will help for the fishermen to protect the environment, as well as it will help them to get good number of good catches in different species. If you see that right bottom, you can find out large groupers coming, and they are living in this particular area. There are different type around. There is one is reef wall and a square type and triangular type. This artificial reefs are deployed. They provide not only protection, they help in actually breeding, and particularly groupers and many other large fishes are growing in this. So that way we are modifying this marine environment, and also we are reducing that trawling operations. Some of the places it is asked by the fishermen themselves, due to reduction in fish catch, they asked us to put this one. So some of the places people have put this due to trawling operations, that bottom environment is degraded. So by doing this one, we can revive this bottom environment. It will stop the trawling, as well as after the deployment, you can go for a long line and other fishing methods. You can collect a lot of fishes in this reef, that is artificial reef areas.
Sir,
So
one
now,
question.
yeah,
Sir, I used to know this in artificial reef, like you said that, and I have seen in slide that there is a artificial this reef is developed near Odisha. So
yes,
can we develop the artificial reef where we can do the like coral reef as it is there in the
yeah,
area?
yeah,
I
it
think
is
is a natural reef, but artificial reef, can we harvest the corals and all that?
yes, yes, we can actually develop, and we can paste in that small pieces of corals, and we can develop. This is very much possible. We are trying 3D printed artificial reef in Andaman Nicobar Islands also. We
Okay,
have a
thank you.
project 3D printed artificial reefs that we are going to test it with some 0.5 meter height and 0.5 meter width. But other this one we can develop. There are so many modules we can make, and we can develop very much possible. This coral reef development is very much possible, but it is a slow process. But in clear waters we can develop.
Thank you, sir.
And apart from that, we are doing that seawater quality monitoring in Andaman Nicobar Islands. This is related to today's talk also. Apart from biotechnological aspect, this is pollution monitoring. More than 40 parameters we are studying. Particularly our part, this this work is being carried out by in entire Indian coast. But for our NIOT, we are doing it in Andaman Nicobar Islands. Many other institutions in India, they are doing in different coasts in different states. Andaman Nicobar Islands, more than 8 stations previously we are studying. Now we are having that is transect in many other bays in Andaman Nicobar Islands. More than 40 parameters we are studying, apart including physical, chemical, and biological parameters. And also these pollution parameters like coliforms and other bacteria, total viable counts and all. And this, based on that, we have more than 20 years of data. From 2000 onwards, we are collecting. This data is developed as a database which is available through other public use, and we have through INCAIS, Indian National Center for Ocean Information Services. They are having this data, and this is being given by another institution, National Center for Coastal Research. They are actually concentrating on coastal area research. They are giving the project to us. This is one of the ministry institution only, our NIOT sister institution. So this is actually being done for past 20 years. We are collecting this data, and we have published many papers related to pollution monitoring. Apart from that, we are doing microplastic studies also. So many microplastic studies. How this microplastics being taken by the zooplankton and how they are dying because of non-digestion of this microplastics, and what kind of microplastics are available in West Coast, Arabian Sea, Bay of Bengal, even in Andaman Sea. All those things we have studied. So they are also found out, these microplastics also found out from the fish tissues, particularly muscle and other gut. They have a lot of microplastics. So if you see the Arabian Sea, we have studied all these west coast, and also Andaman Sea also we have studied all the coast. There are three different kind. This particularly, some are pieces, some are small globules, some are thread-like. So these are all very small, very small, and you have to use microbes, microscope for studying all these things. They are becoming nuisance for our open ocean conditions. Then apart from that, in different, not only deep sea, other bacteria, different bacteria also we are studying. Even this LDPE degradation, as I said earlier, these are all plastic degrading bacteria we are studying that. This is the kit we have produced actually for that microbial, that is multiplex PCR kit we have identified, and we have developed in Andaman Nicobar Islands. Now we have transferred these kits to other people. Then apart from that, there are lot of that microbial studies we have done. I'm not going to explain all those things. This is about ectogen biosynthesis, and from deep sea bacteria, how we have transferred this gene to other bacteria and transferred this technology. This is another L. asparaginase, as I said earlier. This also gene cloning techniques we have used. And this one, other glycine betaine biosynthesis gene. This is one of the osmolate, and it is very deep sea bacteria. From deep sea bacteria, we have collected this betaine, glycine betaine. Actually, previously you asked the question, these are all the things. So this is Trihalus, another deep sea bacteria we have taken. This is particularly from the actinobacteria, Streptomyces quincunlensis. This bacteria which are producing this particular compounds. For that also use deep sea. Then apart from that, there are some mangrove-associated melanin pigment producing bacteria also, which is used for different purposes, this melanin. Then we deployed fish aggregating devices in different places in Andaman Nicobar Islands. Particularly, these are very well used by Lakshadweep people. Lakshadweep people, they are doing long line fishing, and they go. We have deployed more than 28, that is deep, the 28 FADs at depth of 1,200 meter maximum, 1,500 meter. So they can able to go to the site and they can collect lot of fishes. Even one FAD can hold fishes around 5 kilometer range. They can collect. So Andaman Nicobar Islands, it is different places we tried. Some of the places it was very successful, but Lakshadweep it is most successful, and still they are using that this FADs. So we last part we are coming. So recently we have developed ballast water treatment technologies. You know that when the ships are traveling through oceans, and they will take particularly oil tankers and all, they will take a lot to stabilize this ship. They will take a lot of seawater from particular area and for stabilization, and they will transport to that next area before filling that oil, or oil tanker oil or something like that. Then when they are releasing that particularly oil, or then along with that they may release lot of this seawater from that ballast water. So the seawater will contain lot of organisms. So there are organisms which will go and establish in different places, and it will create some bioinvasion crisis in different places. So through seawater, there are so many organisms where actually traveled to different destinations all over in the world, and some of the temperate species started growing in that tropical regions and creating and destroying that local species, some of the places. So this is one of the very important study. Now for that in India, this is the first ballast water treatment. So every ship should contain ballast water treatment system. So they have to remove either they have to kill. Some of the countries they have developed that is rules and regulations. So all the bacteria and other fishes or any other organism which is coming through this ballast water, the eggs and larvae coming through ballast water should be removed either by chemical method or something like that, electrical method, and so many other methods. But IMO, International Maritime Organization, they have given guidelines what is the ballast water content should be, what should, how it should keep that, maintain that as per their rule, like that. So for that purpose, so we have developed this facility at that is at Pavanji, that is Andhra Pradesh. This is the only one facility in India for treatment, because this every fish ships, they should have the certification for their ballast water treatment facility. That certification can be now being done in other countries, Singapore and other places, this ballast water treatment test. But in India, we are the first institution where we are going to test their facility. They'll bring their test facility to our system. We will use different bacteria and different phytoplankton and from 3 different group of organisms, group of phyla, 5 different species we have to maintain. So we are doing all those things. For that, we are maintaining culture, that is algal culture facility also. And apart from that, we have to treat all these things, and we have control tanks and test tanks. From when we are testing through this system, we can find out whether the ballast water treatment facility they are keeping in the that is in their ship, which is working as per the regulations of IMO, International that is Maritime Organization. So this is one of the very important. This one recently this was opened. This is some of the things. These are the feed tanks, control tank, test tank, and sampling points, and we have SOTF. Then apart from that, algal culture facility also we have in this Pamanji stations. So this is overall our marine biotechnological, marine pollution, and other activities which is being carried out by our marine biotechnology group at NIOT. We have a lot of publications and we have a lot of patents, and we are continuously transferring technology to different institutions. For this purpose, we are collaborating with the different institutions. Many of our institutions are other institutions working in these similar areas are working collaboratively with us for developing technology, and also they are publishing many of the publications through international publications through our this one. So we are open to work with any institutions nationally and internationally. So this is all my today's presentation. I hope it will be meeting the today's presentations requirement. Thank you, sir.
Thank you, sir. Very good presentation.
Very good. Yeah.
If there are any questions, I can.
Thank you, Dr. Vineet. I have a brief overview about your activities, what you are doing at NIOT, and specifically Deep Ocean Mission Vertical 3 related to marine biotechnology, marine microbial technology. It's a very lot of observations, a lot of research activities, and also new product, new patent, and so many activities are undertaken by NIOT, as well as the Deep Ocean Vertical 3. So now this is open for discussion. Any of our participants wish to ask any questions or clarifications, and you are most welcome.
Okay, sir. This in Andhra Pradesh, I have seen in the slide that they have developed one ballast water treatment plant.
Yes.
Actually, I have sailed in a ship, a lot of foreign-going ships as a captain.
Okay.
So we already have a treatment plant on board in many of the new ships. So therefore, we are using our own treatment plant.
Okay.
What is the commercial use of this plant which was started in the Andhra Pradesh? Is there any commercial use? Because ships are using either they are doing empty refill method and flow through method, or they are using on treatment plant. Then what is the utility, or it is only for the research? Can you
actually this is for testing and certification purpose. Many of the people already they got certification and they are already using it. And there are many new ships in the Indian Ocean region. They are being built. They have to get the certification for that they can come to that. Apart from that, research purpose also we can use this facility is for not only for ballast water, and we can use it for many other purpose also. Research. Apart from that, this ballast water testing, it is very importantly our ministry is working on that for giving the certification along with IMO. So even the existing, some of the people they want to again test it their facility. Some people are developing again some of the ballast water treating equipments, treatment equipments. They can come and test it here, because this kind of facility is not available in India or surrounding places. You have to test it in either in European this one and other places. But this is the first facility in India which will helpful for testing here itself. Actually, most very importantly, many of this ballast water treatment facilities are tested for temperate waters, not tropical waters. This is the only facility available for tropical waters.
Yes, sir. Thank you, sir.
Excuse me, Dr. Vineet.
Yes.
I appreciate your very elaborate presentation. But this ballast water treatment, who is going to certify in India? The authority of certification in India.
Here, along with the Ministry of Shipping, along with Ministry of Shipping and the Ministry, our Ministry is working on it.
Okay. Any standards are being stipulated?
Yeah, it is there.
Available, already published?
It's already, actually, it is IMO has given guidelines. International Maritime. We have to follow the same methodologies and same guidelines. With that only we can certify. We have to maintain particular that is number of species, and also that even the concentration or number of
yes, CFU in some milliliters of water. That is colony forming units of bacteria and all that.
Yes, microalgae or any other phytoplankton, we have to maintain this 10,000 that is numbers per ml, something like that, in very high quantity. We have to maintain. With that only we have to certify that this system is, whether that system is entirely killing or it is actually, it should not entirely kill. It should keep minimum this many numbers. Because this will not go for non-targeted organisms. That is the main motive. Any other, sir?
So this FADs, I was really surprised to hear this FAD which attracts the fish. So in this case, we can install these FADs all along our coast, no?
Actually, it is being done in other countries, or our even fishermen doing it for very long time. Actually, even any floating material which is, whether it is ship or any our even data by anything, will attract because some curiosity, something like that, that fish will get attracted to that. See, previously in Kerala and all, people will go and put their coconut leaves in bundle. They will put it in one place, and after 1 or 2 days or 10 days, they will go near to that and they can fish. They'll get a lot of fishes like that. This actually, what happens when we put some these FADs, smaller, it will create smaller ecosystem. Algae will develop to eat that. Smaller fish will come to eat the small fish. Little bigger fish will come. Like that. See, in the surrounding, at 5-kilometer range, you will find bigger tunas from the FADs, and nearby you will get smaller fishes. There are hundreds of thousands of FADs deployed by Chinese and Japanese people. They are using it very well. Lakshadweep, they are using it.
Yeah. What about in India, East Coast and East Coast?
East Coast, we have, I don't know, we are not using it mostly. Lakshadweep, they are using it for, they call it as fish banks, because they go near that, so they can, like money they can collect. Because the thing is, they will actually aggregate the fishes in places, so you don't need to search for fishes. In particularly, if you are using this with the potential fish forecasting, that PF set this thing, along with this the FAD, you can use it very effectively.
Okay, is there any plan to talk with the ministry and to deploy it on the East Coast?
No. Right now, if on requirement basis, we will be able to do.
Okay.
And the main thing is, NIOT can, as per requirement, we can deploy in even. We are deploying the different kind of our database. Same way with the lighting conditions or with the lights, that marker lights. During night time, it will be visible. That lights will visible. Red color light will be there, so that that boats can avoid that particularly. They can only fish near to that. They can. What happens when we are putting this kind of things? People will tie bigger boat in that, so there will be. It will come out actually. Actually, this is tested for some that is some particular wave loading conditions. So there will be chain rope below that some 100 meter or something. So people cannot go and cut that chain rope. Below that HDPE, that
So it
bottom at 1,500 meter, there will be big 1 or 2 ton weight will be there. That will be, so this top one is floating. That is tested in
so
this
some
one.
of them is secured to the
Yeah,
seabed, is it?
seabed, yes.
Acha,
These
okay.
all are secured
All
to
are
the seabed.
secured to the seabed.
Yes,
Okay, okay.
there will be swivel in between. There, it is specially designed actually.
Okay.
Otherwise, it will that in between swivel will be there. Even if it is rotates, it swivel will take care of that. This one. So up to 100 meters, there will be steel wire rope, surface to bottom. Otherwise, people will go and cut that. They cannot cut the steel wire. Below that is the HDPE line. Then big HDPE rope will go up to that bottom of that this one.
Till the
Sir,
seabed.
this
Okay.
FAD, what is the basic principle of FAD? This fish gets attracted with the light or with the color or of the boy, or what is that?
Actually,
Can
it
you
is
explain something?
there are different things they are telling, sir. Actually, sometimes light, if you are putting in the below that that system itself, they will get attracted. Even appendages, some kind of nets or something you are tying below that, it will form a that creates small micro ecosystem, so they will get attracted. Even ship, if you are keeping it for sometimes 1 week or something, below that it will act as a feed. So this, so what happens? This tuna and all, they will migrate in a faster way. So if you are having some 10 to 15 FADs, uh, that same micro that is tuna schools will travel one FAD to another FAD. They'll move in the entire distance and come back. So this using it. If you are going for one school of tuna now in one FAD, after 10 days they will find out another FAD near to that, because they know that they will get smaller fish for eating that. So this is being used in many other countries. We are particularly these island nations in Pacific. This thing, they are using it very well. This one, China and Chinese, Japanese, they are using.
So the fisherman community, if they want to use, they can come and approach you,
Yeah,
is it?
definitely. They can approach. And the thing is, we can deploy. It has to be maintained by them. Once in a year, they have to check for this any rope they want to change or cleaning all these things. Once in a year or once in 2 years or something. It can, if you are not touching it, will go up to 4 years. After that, we have to maintain it.
Okay, okay. Thank you, sir. Thank you for the information.
Regarding this, when you are applying this device on the coral reefs,
Hmm.
whether it is not disturbing their ecosystem.
No, we are not putting in the coral reefs, no. We will be deploying in the offshore condition, the 1,500 meter depth or something like that, not in the coral.
I'm
Coral
sorry,
reefs
one of
is-
the sites you mentioned it can be deployed in corals. Okay, no,
No,
problem.
that is artificial reefs. That is different.
Yes, that's what.
That is artificial reef. It can be deployed near the coral reef, not on the coral reef. We can develop artificial reef as a coral reef.
Okay.
FAD is different.
Okay. Thank you, Dr. Vineet, for your telling your valuable time with us and accepting our kind invitation
Yes.
and sharing your experience and expertise, and also the research activities which was taken at NIOT as well as the Deep Ocean Mission project. I'm sure some of our IMU faculty and researchers will approach your university to join collaborative some research activities or projects.
Yeah,
Please
already
extend your help us.
MOU. Already we
Yeah.
have MOU. We will renew it with. We want to have a meeting, so we will do it.
Yeah, please, you can extend your support for us also to develop the center. Thank you very much.
Yeah, thank you, sir. Thank you.
Thank you, sir.
Thank you, all
Thank
of
you.
you.
Thank you. Very interesting talk.
Yeah, thank you.
Yeah, now we have the session, and for that, I am very happy to welcome Professor Chandravadini Devadasan, Rector, Trigonamala Campus, Eastern University, Srinagar. Welcome you, madam.
Welcome.
Yeah, I would like to give brief introduction about madam. Professor Chandravadini Devadasan is a rector and a researcher at the Trigonamala Campus, Eastern University, Sri Lanka. She has more than 3 decades of teaching, research, and administrative experience at Trigonamala University, Eastern University. Her research focuses on aquatic pathology, fish microbiology, immunology, and fisheries. Dr. Devadasan has published very good research articles and also published books. She has contributed to national and international projects as a project advisor or project coordinator. She actively involving with IMU in collaborating and to start some maritime research at Eastern University, and the efforts are underway, and hopefully we will review it, and soon we will have another activity. Already IMU and Trigonamala Campus has a collaborative conference which was held in September, October, and it was very successful conference, and followed that we are organizing this webinar, and we will organize many in future. So she also had very good positions like including acting Vice Chancellor at Eastern University, and has received many awards and both in academic and research contribution. Today she is going to deliver a talk, Marine Pollution and the Effects on the Marine Life, with a special reference to South Sea. Now the platform is to you, madam.
Okay, thank you very much for your brief introduction. Now this is the topic of the given to us today, the marine pollutions. Our Dr. Vinit Kumar, actually basically this the topic we selected for this seminar because the marine biotechnology. We have to how we are prevent or control this pollution, the marine pollution. So now the present days, the technology, marine biotechnology is one of the important tool to prevent this poisonous or toxic pollution in the water. So that's Dr. Vinit Kumar is very extensively presented this talk. Is very it's very useful, and it is I'm proud to say it is there are a lot of the bacteria they identify to biologically control these pollutions in the sea. So the marine pollution, oceanic pollution is commonly, it is the chemical waste and the trash waste they get into the water body and make the changes in the biological process of the marine life as well as the marine ecology. Usually you see that more than 80% of the pollutions and the marine pollution occurred by the land-based resources. So the sources of the pollution get into the polluted material, so we can divide it 2 main ways. One is a land-based solution as well as a sea-based solution. Land-based pollution commonly it is the most detrimental effect on the bays and the lagoons lagoons and estuaries there. But the open ocean is common is sea-based resources like oceanic dumping and offshore spills and the pollution, oil pollution. So the chemical pollution we can be divided many ways. In the metal pollution, it's commonly we call the nutrient pollution. It lead to some algal blooming, and that lead the fish death and the marine animals death. So it's a common scenario, common things is going on in India.
Madam, excuse me.
Yeah.
Do you have any presentation?
I have a presentation. I am presenting now.
No, we are not able to see, madam.
I said share my slides?
To us,
I already I share my slides. Maybe setting over there.
oh, that we cannot be able to see.
I have share the slides. Any problem? I share the slides.
Your PPT is not visible.
It's not visible.
Can you share it now?
Wait. Again, I will share that entire screen.
Yeah.
Window and window, entire screen. Window. Can you see now?
No, madam.
So what is earlier also I can't see your slides when you Mr. Dr. Vinith Kumar presented. I cannot see that. Some problem in that.
Now we can
is
see
visible.
something. We can see something. But it is not slide. It is not slide.
So what is the problem? It's the share. They say this. Wait. I try again.
You can. Sugandhya. Sugandhya will go on.
Can you see now?
It's in the percentage. Can ask for percent here.
She presented.
Pardon? Can you see? No?
Oh, madam, it is the same.
Something else is there in your screen, not
No,
the
you're
presentation.
sharing the wrong screen.
The screen is.
அதுவும் பண்ணலாம் ஆனா அவங்க அனுப்பிச்சு பண்ணாங்க கரெக்டான ஸ்கிரீன் ஷேர் பண்றது ஈஸி டார்கெட் பண்றாங்க டார்கெட் கேட்கறாங்க இல்ல அவுட்
Yes, to join.
சம்டைம்ஸ் லாக் அவுட் பண்ணிட்டு வந்தா ஷேர் ஆகும் அதனால லாக் அவுட்
Ah,
Yes, now we can see the slide. At least we can see this slide.
அது இதுல full screen வராதோ?
Hello, can you see them?
Yes, madam. You can see them screen.
Right.
Yes,
Okay.
yes.
Right. How can you go down? So there's a two type of the chemical pollution as well as that other trash pollution. So in that, when you look at the earth, wait.
Full screen வரலைல.
Madam, your fifth slide is coming.
Okay, so there are countries like this pollution is a common in the developed countries like USA and China, Japan. They are worse offenders, and in terms of the littering of the
Madam.
pollution. Yes.
Just madam, 1 minute.
Can you switch on the slideshow, madam?
Can you see that one?
Yeah, the slides are visible. The slides are visible, but can you just press that from beginning there? If you press it, it will become full screen, so it will be much more visible.
Wait.
This side வரல.
Wait. Okay. Can you see now?
We are seeing the slide, madam, but not the full screen.
I could see the full screen, but you cannot. Can you see the full screen?
No, madam, we can't see the full screen. Ma'am, it's okay, ma'am. You can continue. It's okay.
Okay, right. So today's the problem is the pollution in our oceans. It is the common problem, the possible impact of these pollutions to totally our marine habitat, aquatic habitat are modified, coastal marine habitat as well as the mangroves adjoining area in the located in the adjoining areas of the marine habitat, coral reefs, and the seagrass bed and estuaries and the lagoons. The country like a whole earth, we look at the earth, there will be a possible the most recorded problem in the developed country oceans, in the Pacific Oceans, and the South Sea and the southern part of the seas are mostly polluted. And my studies I done in the Southern Sea, how far this pollution, the polluted material affect the growth of the marine life, and the Indian and Indonesian countries.
Sorry for the disturbance. Some network problem it seems. I'm trying to call her, but it's not picking up. Please wait for a few minutes. Hello? Anybody can tell whether it is, you can able to hear their voice? Hello?
No, sir.
Oh, okay. You are not able to hear their voice. Madam, nobody able to hear. No. Actually, you are not in the thing. You have to please rejoin, madam. I think you are not in the either. Yeah.
Do it then.
Yeah. No. You are not. Is it possible to reconnect now? Yeah. Oh, okay. But no, nobody is not hearing. No. You can able to join, madam? Oh. Okay. Sir, we are, sir, can you hear me? Hello?
We can hear you, sir.
Yeah. Sir, there was some problem in the country at Sri Lanka. You know the difficulties. There's some network problem there. They are not able to connect now. So I think we can stop it, and another day we will try to organize this. Anyhow, we are thankful to all of you for kindly accepting our invitation and taking part in this webinar. But due to some technological issue, we are unable to complete this event, and most probably later we will do it. And I'm extremely sorry for the delays and the inconvenience caused you all for waiting for a long time. So thank you very much for all of you. And my special thanks to Dr. Vinith Kumar for spending his valuable time and also sharing his experience with us. Thank you very much for all of you.
Thank you.
That certificate will be sent to you, sir, after this. We will send the email.
Okay, thank you, sir.
Thank you. Thank you very much.
So sorry, sir. Sorry for the inconvenience. Yes. Now you can leave the business.
Textbooks and all.
Sir, we are closing now. Please.
Close பண்ணிடுங்க.